All right, so we're going to do some problems, and we'll have the same format as last time. Um, get yourselves in partner groups or bigger, up to maybe 4 people if you want, and work together. Talk, definitely talk, and puzzle through these problems. And I'll go first, and then Nick will go second. So this is the first problem. If it's helpful, here's a data set that could be the result of those mutations, right? Where this is sequence 1 and sequence 2. And I know the ancestor. And this would be the probability of it. This is a hard problem, especially with 3 sequences, but try your best. Anyone have an answer? Getting close to an answer? All right, we don't have many minutes left. Let's maybe just walk through it together. What would the tree need to look like, the gene genealogy? With 3 sequences, there's only really one shaped tree, right? And what sequences would need to be over here? Sequence 1 and sequence 2, yeah? And sequence 3. And then where would the mutations need to be on the tree? Up there, yeah, exactly, right? All right, and so now that's good. We can just apply our same thinking here, right? There's a couple of tricky things. The first one is that when there's n sequences, then I would have n theta over 2 rate of mutation, theta over 2 for each of them, and n choose 2 rate of coalescence. In this case, I would have 3/2 theta and 3, right? And these would be the relative probabilities of coalescence and recombination. I mean, mutation being the first event backwards in time. And so instead of theta over theta plus 1, if I took the ratio of this over that plus that, I would end up with theta over theta plus 2. Right? So the first part has probability 2 over theta plus 2. And then I have to have a particular one of the 3 possible coalescent events. And then I have to have theta over theta plus 1, and that factor of 1/2. And then another theta over theta plus 1, and that factor of 1/2. And then 1 over theta plus 1. I multiply those things together, and I have the probability of those data under the infinite sites mutation model, standard coalescence. Yep. You could, but then you would need to know the total distribution of the total tree length. Yep. And you do some integrals and stuff, sure. Yeah. But not, I wanted to know the particular pattern here, right? That would be just the number of sites. Yeah. Yeah, exactly. So this term here, that 3 there, includes 3 different possibilities, right? Yeah. And if there were 4, it would be 6 and so on. Yep. So that's what would happen if you sequenced some data and found very few polymorphic sites in a sample of size 3, and you knew the ancestral state, and fed that into a coalescent program, and had it spit out the likelihood, or a likelihood surface. Anyone with a computer open could make this product in some mathematical program and graph it, and that would be the likelihood function for those data. And you could make inferences about theta under using standard statistical likelihood theory. All right, so I should stop there and let Nick take some time.